Hello, my dear students. Welcome to another lecture. And in this lecture, we are going to talk about the structure and functions of neuron. Now, we're going to talk about a lot of things about neuron. And in this lecture, we will actually divide this into a couple of parts. And in this part, we will mainly focus on the structure of the neuron and what are the different parts of the neuron and what is their exact function. Let us start. So we will be talking about the structure and the functions of neuron. Now before that, let's have the basic introduction to the neurons. Now what exactly is this neuron? Neuron is actually also called as a nerve cell, and it is the basic functional unit and structural unit of the nervous system. Now nerve or a nerve cell, it is actually an excitable cell that is electrically stimulated. It is electrically excitable cell that is meant for processing and transmitting the information by the electrical and chemical signals. Yes, so neuron is an excitable cell that actually processes and transmits the information by the electrical and chemical signaling. Now, as I've already told you that these neurons are the core components of the nervous system. And when we talk about the central nervous system, in brain only, there are about 86 billion neurons that are making our brain. Okay, and we have these neurons, when they transmit information from one part to another part, they transmit information from, for example, from one neuron to another neuron, and the junctions that these neurons are doing that, they are called as synapses. Okay, now let's talk about the structure. We will mainly focus on the structure and the functions of neuron. And here is the structure. Now there are different types of neurons, but there are some typical structures that are common in all these types. And we will be talking about these parts of the neuron first. Now, the first thing, because as this neuron is a cell, so it has those cytoplasm, nucleus, and other cell organelles present inside, like every other cell in the body. Okay, now the part of the neuron where all these things are present, mainly the nucleus and the other cell organelles, that part is this round part of the neuron, and this is called as the cell body, because every cell has a cell body and where the nucleus and other cell organelles are present. Likewise, we have the cell body of the neuron where there is this nucleus present and all the other cell organelles present. Now what are the very important cell organelles present inside? We will be talking about that in the functions. Now the second thing is, from this cell body there are arising these processes. There are these cytoplasmic extensions that are arising from this cell body, these branches. And these branches, so this is the cell body, and these branches, they are called as the dendrites. They are called as the dendrites. Okay, and from the cell body there is another process that is arising. You can see this long process arising from the cell body, and this part is called as the axon. We will be talking about all these parts in detail, and the terminal part of the axon is again these branches. So this axon is actually dividing into several branches, and these branches are having a broad end in their terminal part. So these branches here, they are called as the axon terminal. Very straightforward that these are the terminal part of the axon. So axon terminal. So we have the structure of the axon divided into four main parts. The first part is the cell body. Like every other cell, it also has the nucleus and other important cell organelles. And all these cell organelles, this nucleus is present in the cell body. The second thing, from the cell body are arising these extensions, these branches, these processes, and they are called as the dendrites. The third thing, from the cell body itself, there is arising another branch and another process, and this is called as the axon, and the terminal part of this process of the axon is called as the axon terminal. Now let's talk about all these things one by one, and we will talk mainly focus on the functions of all these parts. Now, the first is cell body. Now, cell body, as I've already told you, it is very, very important because it is the control center of the neuron. It contains the control center of the neuron, and that is the nucleus. Okay, now here is the cell body. You can see here is the cell body, and this cell body is actually containing the nucleus, and this is the euchromatic nucleus. Okay, euchromatic nucleus means it is active in that process of transcription. There is active transcription going on inside because it is involved in the formation of many proteins. Okay, now what are the important cell organelles present inside this this cell body? You can see here from the cell body are arising these different branches we will be talking about next. These are the dendrites. So this round structure right here is the cell body, and this cell body, this is the cell body here, cell body, and this cell body is also called as soma. So soma is the another name for cell body. Now, what are the important cell organelles? The first important cell organelle are these structures here, and these structures, like every other cell in the body, this neuron also has the cytoskeleton inside. And cytoskeleton here in the neuron cell body, we call it as neurofibrils. Neurofibrils. Now, neurofibrils, the cytoskeleton mainly contains that microfilaments, for example, the actin, or these neurofibrils. Why they are important? Because they are very important in providing the structural integrity to this neuron cell body, or structural integrity is maintained. Structural integrity of the neuron is maintained by the neurofibrils. Now, cytoskeleton also contains the microtubules that we will mainly focus in the axon part because they have got a very important function there in the axon. Now, the first is neurofibrils. Now, the second part here is this round, very important structure is this round big structure, and this is called as the nucleus. And this nucleus is the control center of the neuron. Okay, now as I've already told you that it is euchromatic nucleus because it is involved in that active transcription processes. Now the third thing, because this neuron has to use a lot of ATPs, a lot of energy for doing all the metabolic processes, and all these things, this energy is provided by an important cell organelle. We all might be aware about that, and that is our mitochondria. So mitochondria is also present there inside the neuron cell body. Now remember, the mitochondria is not only present in the neuron cell body, it can be present in the axon up to the axon terminal, because we need that energy throughout the length of the neuron. Now, another important thing is this fourth structure here. You can see these tubular structures, and these tubular structures are the microtubules. So, number 4, we have microtubules. Now, these microtubules are actually made up of a protein that is called as tubulin protein. We have alpha tubulin, we have beta tubulin. Okay, about these microtubules, I've already told you that they are performing the very important functions in the axon. So we will be talking about the microtubules in detail in the axon part. Now, the fifth part is right here. Okay, these structures here, and these structures are the rough endoplasmic reticulum. Okay, now rough endoplasmic reticulum, as you all might be aware, that these rough endoplasmic reticulum is involved in the synthesis of proteins because they contain the granules on their surface, and those granules are the proteins, these ribosomes, and ribosomes are the protein factories inside the cell. So these rough endoplasmic reticulum is involved in the formation of proteins. There are different types of proteins that can be synthesized inside the cell body. Okay. Now, there is another important structure inside this neuron. There are the stacks of rough endoplasmic reticulum and the ribosomes. And the stacks of the rough endoplasmic reticulum and the ribosomes, or the clumps of this rough endoplasmic reticulum with the ribosomes, that is called as the Nissel's granules inside the neuron. That is called as Nissel's granules inside the neuron. Okay, and then there is this last part. There can be free ribosomes as well. So these are some important, yes, one more important thing is there is another structure that I will write here, and that is called as Golgi apparatus or Golgi body. Now, Golgi, we all know that it is involved in the processing, packaging of the proteins that are synthesized in the rough endoplasmic reticulum. Anyway, now we have seen that these are the important parts that are present there inside the neuron cell body. Now, what exactly is an important function of the neuron cell body? Because it has all these structures. It has the nucleus, it has the rough endoplasmic reticulum or the Nissel's granules, it has the Golgi apparatus. Definitely the very important function of the neuron cell body is the synthesis of proteins. Its function is the synthesis of proteins. Synthesis of proteins. Now, the thing is, what type of proteins are important that are synthesized inside the neuron cell body? The first important thing that should come in your mind when we talk about the synthesis of protein inside the neuron cell body should be the neurotransmitters. Because when this neurotransmitters, when this neuron has to pass the information, or it has to communicate with another neuron, it is actually done via the chemicals, and those chemicals are called as neurotransmitters. Now, those neurotransmitters are synthesized inside the neuron cell body by the process of this transcription followed by the process of translation. The second thing that can be inside here can be the membrane proteins. Membrane proteins, and we all know from the structure of cell membrane that cell membrane contains very important proteins inside. For example, there are different channels in the membrane of this neuron. There are the ligand gated channels, there are the voltage gated channels, there are the leaky channels, and all those channels are meant for the transport of ions across the cell membrane, and those membrane proteins will be formed inside this neuron cell body. The third important protein can be the structural proteins. These membrane proteins can be the structural proteins, but the structural proteins that are used inside the cell. For example, I have already told you that there is an important structure that is called as microtubules, and microtubules are made up of these tubulin proteins, and for example, these tubulin proteins has to be synthesized. They will be synthesized inside the neuron cell body. The next important thing that can be synthesized there are the enzymes. Okay, enzymes that are utilized for different metabolic processes inside the neuron cell body or inside the neuron, those enzymes can also be synthesized here. Okay, now you all might be aware the process of protein synthesis. For example, inside the nucleus from DNA we are forming that mRNA, and that process is called as the transcription process. And then this mRNA will go out of the nucleus, it will go to the ribosomes or the rough endoplasmic reticulum. Then this mRNA will be translated into a protein. Okay, and that process is called as the translation. And then these proteins, for example, in order to shift these proteins from this neuron cell body to the axon terminal, we have these structures called as microtubules. For example, you can see here, we have these proteins in the form of vesicles that are coming from the ribosomes or the rough endoplasmic reticulum, going to the Golgi apparatus for the processing and packaging. Then these vesicles are moving down via the microtubules and they are transported to the axon or axon terminal. Okay, so coming back here, we have the first important function of this neuron cell body, and that important function is the synthesis of proteins. Now, the second function, very, very important function, that this neuron cell body is responsible for generating the graded potentials. Graded potentials. Now, these graded potentials we will discuss when we will talk about the dendrites, because dendrites are mainly responsible for generating the graded potential. We will talk that in detail when we talk about the dendrites. Now, graded potentials, you have to remember, can be EPSP, that is excitatory postsynaptic potential, and IPSP, inhibitory postsynaptic potential. Now, what this exactly is, we will talk about in dendrites. Now, one more important thing before we go to the next part, that is dendrites, that there is a process called as chromatolysis. Chromatolysis. Now, what happens in chromatolysis? Chromato is made up of two of these words. Chromato means color and lysis means breakdown. So what did I tell you? That the Nissel's granules are the stacks, are the clumps of the rough endoplasmic reticulum and the ribosomes. Now, what this chromatolysis is, for example, this is the neuron, and this is the neuron, and here is the cell body, the axon, and here is the axon terminal. Okay, now what will happen is, for example, during any injury to the neuron, severe injury, for example, there is the cut in the nerve fiber in the axon. There is the cut, and we have to repair this. We have to repair this. The cell body receives the signal that the impulse is not transmitted from the cell body to the axon to the next neuron or to the target or effector organ. Okay, now what will happen? This neuron will start the repair mode. It will start the repair mode, and what will happen? There will be the breakdown of these Nissel's granules, and these Nissel's granules will be dispersed throughout the cytoplasm. Okay, and what will happen is this nucleus will shift to the periphery. It will shift to the periphery. It will shift near to the axon. Okay, where the axon is arising from the cell body, and it will move, suppose for example here, and there will be swelling of the cell body. Swelling of the cell body means it is involved in a lot of metabolic processes. It is involved in a lot of the formation of a lot of proteins, the proteins that has to be involved in the repair and regeneration of these neurons. Okay, now when we talk about the nerve injury in the peripheral nervous system, we have the Schwann cells there. The Schwann cells help the repair of the neuron in the peripheral nervous system, but the repair in the central nervous system is very, very difficult because the oligodendrocytes there in place of Schwann cells do not help the neuron in the repair and regeneration. Okay, this is very important. So this process is called as chromatolysis. Chromatolysis is simply the breakdown of these Nissel's granules. There will be the swelling of the cell body. That means the cell is not in the mode of neurotransmission. It is in the mode of repair and regeneration, because it is synthesizing the lot of proteins, those proteins which are very important. For example, it will start synthesizing the structural proteins that I've told you. It will start synthesizing the different growth factors that are very, very important for the repair and regeneration. So that process is called as chromatolysis. So in short, what is the cell body? The cell body is the main important part of the neuron, and it contains the nucleus, the control center of the neuron, and it contains all the cell organelles that are very, very important. For example, in the formation of proteins that are very, very important in maintaining the structural integrity to the cell body. For example, neurofibrils. It contains the organelles like mitochondria. Mitochondria provides the energy to the cell. Okay, and this apart from the formation of proteins in the cell body, it is also involved in generating the graded potentials. Now, what are these graded potentials? We will know in dendrites. Now, here are the dendrites. Now, dendrites, you can see, as I've already told you, that this is the cell body, and from the cell body are arising these processes, and these branches are arising from the cell body. These are the cytoplasmic extensions right here, and these branches are making, for example, it is the multipolar neuron. It is making the appearance of a tree, because these are the different branches that are arising from the cell body. Okay, now these dendrites are very, very important, because these dendrites are the receiving ends of the neuron. Receiving ends of the neuron. That means if this neuron has to receive the signal from another neuron, for example, here is a neuron, and this neuron has to give the signal to another neuron, and this signal by this neuron will be received with the help of these branches, that is called as dendrites. Okay, and these dendrites have actually little extensions. These dendrites also have the little extensions. For example, this is the dendrite. It has the little extensions like this, and these little extensions are called as the dendritic spines. These are called as dendritic spines. Now, these dendritic spines are very important, because these are actually what exactly is the receiving end, because for receiving the impulse, it has to have the receptors, it has to have the channels, and those receptors and the channels are present on these dendritic spines. Now, these dendritic spines, if you can see here in the structure, it has the head, this bulbous part, and then it has this tubular part. This is called as the neck of the dendritic spine. Now, neck actually connects the dendritic spine head with this dendrite, and the head is actually that receiving end. It makes the synapses with the another neurons. Okay, with the other neurons. Now, these dendritic spines are very, very important, because for example, any process of adaptation, any process of learning and memory, we need to have we need to form more synapses with many neurons. Okay, we have to increase the number of synapses. So what will happen? There can be changes in these dendritic spines in the processes like learning and memory. What will happen? These dendritic spines will either increase in the number, or they will increase in the size. And if they increase in the number or in the size, that means that receiving portion is going to increase, that contains the different receptors, the receptors that are receiving the impulse from another neurons. Okay, now this process of increasing the number of synapses by increasing the number or the size of these synaptic dendritic spines, this process is called as plasticity. This process is called as plasticity. Now, plasticity, when we will talk about the synapses and the properties of synapses, we will talk about the synaptic plasticity in a little more detail. It is just the process of increasing the number of synapses with the different neurons that is very, very important for the processes like adaptation, learning, and memory. Okay, now I have already told you in the cell body that cell body is involved in generating the graded potentials, and that graded potentials is generated by the dendrites as well. And so I've already told you that we will be talking about that here in the dendrites. Now, suppose here is a dendrite and here is a neuron. And this neuron has to transfer the impulse. This neuron has to transfer the impulse to this neuron. Okay, and the junction here between these two neurons, that is called as synapse. Please understand this. We will be talking about synapse in detail, but the junction between these two neurons is called as synapse. Now, suppose this neuron, this big neuron here, is after the synapse, after the junction. So this neuron will be called as postsynaptic neuron. This is called as postsynaptic neuron. And this is before the synapse, before the junction, this neuron, this is called as presynaptic neuron. This is called as presynaptic neuron. So please understand this. Now the presynaptic neuron has to transmit the information to the postsynaptic neuron. Okay. Now how it will do it? I've already told you that it is done in the form of chemicals, and those chemicals are called as neurotransmitters. They are called as neurotransmitters. Now, neurotransmitters, based on their function, can be of two types. One is the excitatory neurotransmitter. This is very important to understand the graded potential. That is why I'm telling you all about this. This neurotransmitter can be excitatory, it can be inhibitory. Now, as the name suggests, excitatory neurotransmitter is going to excite this neuron, and inhibitory neurotransmitter is going to inhibit this neuron. The examples are, the best example for excitatory neurotransmitter is glutamate. Okay, if it is releasing glutamate, it will excite this neuron. And if it is releasing the inhibitory neurotransmitter, for example, GABA, what this GABA will do is it will try to inhibit this neuron. Now, how it is doing this? Let's understand this with this diagram here. Okay, for example, we are zooming this dendritic portion here. Okay, we are zooming, and what we find is this is the membrane of the dendrite. This is the outside. This is the inside. Okay, now this dendritic membrane contains this receptors, as I've already told you that it contains the receptors there. Now the receptors can be ligand gated channels. The receptors can be in combination with the ligand gated channels, and receptor can be a G protein coupled receptor. Now what will happen is, for example, here is a neuron, and this neuron is the presynaptic neuron, and this neuron, this is the membrane of the dendrite of the postsynaptic neuron. Here is another neuron. Understand this, please. Here is another neuron, and this will release the excitatory neurotransmitter. It will release glutamate, and this neuron will release the neurotransmitter. This will release GABA. Now, how this is stimulating and how this is inhibiting, we will find out. Now, for example, this glutamate will come and bind on the receptor, and this receptor is, for example, the ligand gated channel. Okay, and it will bind on the receptor here, this glutamate. It will bind on the receptor and it will open this ligand gated channel. This ligand gated channel is usually closed, but it will be opened once a ligand comes and binds. And in this case, the ligand is the glutamate. Okay, now it is ligand gated channel. That means it will be opened only because of the ligand. So what this glutamate will do, it will bind on the receptor, it will open the ligand gated channel, and it will open the ligand gated cation channel. Ligand gated cation channel, for example, most importantly, ligand gated sodium channel. And when this ligand gated sodium channel is opened, what will happen? The sodium concentration outside the cell is more, and the sodium concentration inside the cell is less. So when the channel is opened for the transport of sodium, what will happen? Sodium from outside will come into the cell. Okay, it will come into the cell, it will come into the dendrites. And inside the cell, it will become more and more positive. For example, the cell was at rest before this glutamate came. It was having the resting membrane potential of -70 millivolts. Okay, because we all know the resting membrane potential of the neuron is -70 millivolts. When the sodium is coming inside, it is becoming more and more positive inside. That means it will become less negative from -70 millivolt to -55 millivolt, for example. And this 55 millivolt is the threshold potential. So that means this glutamate, what it does is, this glutamate increases the influx of sodium into the cell, and because of that influx of sodium, the resting membrane potential is going towards the threshold potential. Okay, so that means it is exciting the cell. Now it is easy for this cell at -55 millivolt, it is very easy for the cell to generate the action potential, to generate the nerve impulse. That means what this glutamate has done, it has excited this neuron. by releasing by opening the ligand gated sodium channels. As it is the excitatory potential and it is in the post synaptic neuron, so we call this potential, when the resting membrane potential is taken towards the threshold potential, we call this potential as EPSP, that is excitatory post synaptic potential. Okay, now this is excitatory because we have taken the resting membrane potential towards the threshold potential. Okay, and it is in the postsynaptic neuron, so we call it as excitatory postsynaptic potential. Now, what about the GABA? Now, GABA will come and bind on the receptor. Okay, for example, GABA has the receptor here, and it will open. It will bind on the receptor. Now, what it will do is it will open the ligand gated anion channels, or most importantly, what it will do is it will open the ligand gated potassium channels. This GABA, it will open the ligand gated chloride channels or ligand gated potassium channels. Now, what will happen if it is opening the channels that are allowing the transport of potassium? As the sodium was more outside the cell, less inside the cell, it is opposite for the potassium. Potassium is more inside the cell and less outside the cell. So that means potassium will move from high concentration to low concentration, and this potassium will move from outside to inside. Sorry, from inside to outside. So that means in this case, the positive charges are leaving the cell. The positive charges are moving out of the cell. So inside it will become more negative. Okay, so that means if it was at rest minus 70 millivolt, for example, it goes to minus 90 millivolts. So that means cell is going for hyperpolarization. In this case, it was depolarization, and in this case, it is going for hyperpolarization. That means it is going away from the threshold potential. Okay, it is going away from the threshold potential. To go to the threshold potential, this minus 70 millivolt has to go to minus 55 in this case, but it is going from minus 70 to more negative, that means more minus 90 millivolt. So that means it is very hard for this neuron to generate the action potential. That means we are inhibiting this neuron, and we are inhibiting the postsynaptic neuron, so we call this as IPSP, that is inhibitory postsynaptic potential. Okay, so we have the EPSP, we have the IPSP. Both EPSP and IPSP are called as graded potentials. Now, these graded potentials are either we are exciting the neuron by generating the EPSP, or we are inhibiting the neuron by generating the IPSP, that is inhibitory postsynaptic potential. Now, that is the role of dendrites, how it receives the information and how that neurotransmitter is actually exciting or inhibiting this neuron. Okay, now I have already told you that not only ligand gated channels, there can be, for example, this cell membrane I'm extending up to here, there can be G protein coupled receptor. Okay, there can be G protein coupled receptor, and that G protein coupled receptor, for example, here is the G protein. Okay, when this neurotransmitter, for example, glutamate. Okay, I will give the example of glutamate only here. This glutamate will bind on this receptor, and this receptor is not bound to ligand gated channel. It is bound to G protein, this receptor. Now G protein, what it will do is it will, by certain after certain steps, it will activate the second messenger. It will activate the second messenger system, and second messenger, what it will do is it will activate different protein kinases. Okay, for example, protein kinase A, and what this protein kinase will do is it will phosphorylate or stimulate the sodium channels. It will phosphorylate the, okay, right, I will write it here, it will phosphorylate the sodium channels. So that means it is That means it is stimulating the sodium channels, but not using the ligand gated sodium channels. It will stimulate these sodium channels, and these sodium channels will be stimulated by means of the second messenger system. So this can also be the mechanism of these neurotransmitters, and if we are stimulating the sodium channels, there will be influx of sodium, and that will lead to EPSP. If it is GABA, that will stimulate the second, or that will use the second messenger in phosphorylating the potassium channels, for example. And if we are activating the potassium channels, there will be efflux of potassium, and that will lead to the IPSP, that is inhibitory postsynaptic potential. Okay, so that was all about the dendrites. All you have to know about the dendrites is that these are very, very important in receiving the impulse from another neuron, and it is very important in generating the graded potential, and graded potential can be EPSP, it can be IPSP. One more important thing, that the cytoplasm, the Nissel's granules, the mitochondria, these Nissel's granules can be present in the dendrites as well. This is a very important difference between the dendrite and the axon. The dendrites can contain these Nissel's granules, but the axons will not contain the Nissel's granules that we have talked about in the cell body. Now, here we have the another part of the neuron, that is the axon. Okay, now axon is another branch that is arising from the cell body. And remember that the site at which the axon is arising from the cell body, that part is called as axon hillock. Okay, this is called as axon hillock, and axon hillock is more of a structural name. Functionally, this axon hillock is called as trigger zone. It is called as trigger zone. Okay, because this axon hillock is something where the action potential is generated. Okay, that is very important because we have not generated the action potential yet. What we generated in the cell body and the dendrites, that was the graded potential. We only took the potential from resting membrane potential to the threshold potential. Okay, now that this action potential actually will be generated in the axon, and more importantly the axon hillock. It will be generated there. Now why it is so? Because this axon hillock contains lot of these channels, and these channels are the voltage gated channels. Okay, now the axon, most importantly the axon hillock, it contains the voltage gated sodium channels and the voltage gated potassium channels. Voltage gated potassium channels. Now what will happen, that we have generated the graded potential in the dendrites, that will be taken to the cell body, or we have generated the graded potential in the cell body. Now that graded potential from cell body will move towards the axon. Okay, once it reaches the axon hillock, we have taken, that is the graded potential. For example, we have generated the EPSP there. Okay, and that EPSP we have taken from -70 to -55 millivolts. Okay, -55 millivolts. Once this -55 millivolt will reach from cell body towards the axon, this -55 millivolt will stimulate these voltage gated sodium channels. And because these voltage gated channels, that means they are closed and they will be opened by a particular voltage when a particular voltage hits these channels. And that particular voltage, in case of voltage gated sodium channels, please remember, is -55 millivolt. That is why we call it as threshold potential. That means once the threshold potential is reached, nothing can stop the neuron from generating the action potential. Because this -55 millivolt will reach the axon hillock, it will stimulate the voltage gated sodium channels, and it will open the voltage gated sodium channels. Now once the voltage gated sodium channels will open, what will happen? There will be a lot of sodium that is coming from outside to inside. Okay, via these voltage gated sodium channels. Okay, now this inside the cell, it will become more and more and more and more positive. Okay, so it will reach from -55 to +35 millivolts. Okay, आपने वो ग्राफ देखा होगा. For example, we have taken to the, this is the resting membrane potential, it is minus 70, and we have taken it to minus 55, that is threshold potential, and that was taken by those EPSPs. Okay, EPSPs took the potential from resting membrane potential to threshold potential. Once the threshold potential will be reached, what will happen? There will be opening of these voltage gated sodium channels, and it will generate the action potential. Okay, now it will generate the action potential. It will go from minus 55, for example, to plus 35 millivolts. Okay, and this plus 35 millivolt from minus 55 millivolt, this is called as the wave of depolarization. This is called as the wave of depolarization. Now, once the voltage reaches plus 35 millivolts, these channels will close. These are minus 30 millivolts, usually minus 30 or minus 35 millivolts. So once this voltage which will reach plus 30 millivolts, these voltage gated sodium channels will close, and this will open another channel, and that is voltage gated potassium channel. Okay, and voltage gated potassium channel will open, and it will lead to the efflux of potassium from inside to outside. And what will happen? This depolarization wave, it reached from minus 55 to plus 30 millivolts. Now it will come down. Okay, now it will come down, and this wave is called as the wave of repolarization. Now, we will talk about this action potential in detail. We will talk about the resting membrane potential and action potential in detail in the future videos. But here is just for the understanding. What is the function of axon? Axon hillock is very, very important in generating the action potential. Now, how it is doing that? Because it has a lot of voltage gated sodium channels and voltage gated potassium channels. Voltage gated sodium channels, very important for the wave of depolarization. Voltage gated potassium channels, very important for the wave of repolarization. Okay, so that means we have generated that action potential, we have generated the impulse. Okay, now what will happen? This action potential, this wave of depolarization from axon hillock will go all along the axon, and it will reach the axon terminal. Okay, but remember, the wave of depolarization is going towards the axon terminal. It will be followed by the wave of repolarization. Because wave of depolarization will be because of the opening of voltage gated sodium channels, and the wave of repolarization will be because of the opening of the voltage gated potassium channels. Okay, we will talk about the mechanism in detail in the future videos. Okay, now this wave of depolarization followed by the wave of repolarization is called as the action potential, and action potential is generated in the axon hillock, and it is conducted by the axon towards the axon terminal. So that means no conduction is the second example. So the second function, the first function is generating action potential, and the second is conduction of action potential. Okay, now what is generating the action potential? The opening of a lot of voltage gated sodium channels in the axon hillock. We have generated the action potential. Now that action potential is conducted towards the axon terminal, because we need that action potential, we need this plus 30 millivolt to reach to the axon terminal. It has a very important function there. Okay, so that has to be conducted all along the axon towards the axon terminal, and that is also done by the axon. Okay, now another important function of the axon is the intracellular transport. Now, what is intracellular transport? Suppose we have to transport certain things inside the neuron. We have to transport certain things from cell body to axon terminal. We have to transport certain things from axon terminal to the cell body. Now, that transport is done by this axon. Now, how it is doing that? You can see this green structure inside the axon, and I've already told you that it is the microtubules. Now, what this microtubule is doing there, मैंने आपको बताया था कि microtubule has got a very important function in the axon, and it is very important for the axonal transport. It is very important for the intracellular transport. That means transport of substances from one part of the cell to another part of the cell. Okay, now microtubules is a tubular structure, and this tubular structure is made up of tubulin proteins. Tubulin proteins, and tubulin can be alpha tubulin, it can be beta tubulin. So it is made up of this alpha tubulin and beta tubulin. So that is about the tubulin. But what is important here is the axonal transport, and how this microtubules are doing this. It has got these motor proteins. You can see here, these are the motor proteins. And these motor proteins, there are two very motor proteins that are present there. One is responsible for taking the substances from cell body to the axon terminal, and another is very important for transporting the substances from axon terminal back to the cell body. Okay? Now, the transport of substances from the cell body towards the axon terminal, we call this as anterograde transport. Anterograde transport, and this anterograde transport is done by an important motor protein that is called as kinesin protein. Okay, now the thing is that what type of substances can be transported from the cell body to the axon terminal, or what type of substances can be transported from cell body down the axon. Now, the first important thing that I've already told you is the neurotransmitters. The neurotransmitters will be secreted from the neuron at the axon terminal, but they are synthesized here in the cell body. So from cell body to the axon terminal, these neurotransmitters will be transported by means of kinesin from the cell body to the axon terminal. Then there can be different growth factors. There can be growth factors that are very important for the repair and regeneration of the neurons, and that are synthesized here in the cell body, and those are transported down the axon via these microtubules, and in the microtubules by these motor proteins called as kinesins. The third important thing is any cell organelle. Any cell organelles. And cell organelles, by cell organelles, what I mean by, for example, I have to transport the mitochondria from cell body to the axon terminal, because mitochondria, I've already told you, we need to generate the energy all along the neuron, and we need to transport the mitochondria from the cell body to the axon terminal. That is also done by the kinesin protein. There is another important thing, that is different proteins. For example, the structural proteins and or the membrane proteins. Structural or the membrane proteins that can be used all along the axon or axon terminal. So these structural and membrane proteins that can also be transported from cell body down the axon towards the axon terminal. That is done by the kinesin protein. Okay, now another motor protein that is present there is responsible for taking the substances from the axon terminal towards the cell body. And this type of transport is called as retrograde transport. Retrograde transport. And this retrograde transport is done by another motor protein that is called as dynein. Okay, that is called as dynein. Now what this what this dynein is actually transporting, for example, here is any dead or dysfunctional cell organelle. Abhi maine aapko bataya tha that we need to transport the different cell organelles from the cell body to the axon terminal. Now suppose any mitochondria here, this is the mitochondria, and this mitochondria is now not functioning properly. It has become non-functional. Now what we need to do is we need to degrade this non-functional, this cell organelle. And for the degradation, we need to take this cell organelle back to the cell body. Okay, and that dysfunctional cell organelle will be transported from the axon terminal back to the cell body by means of retrograde transport by these motor protein that is called as dynein. The second important thing, when we secrete the neurotransmitter here, okay, this neurotransmitter, after it has performed its function on its effector or the target, it can be another neuron, it can be a muscle, it can be a gland, whatever it is, it has done its function. Now we need to take this neurotransmitter away from this space here in order to prevent the repeated stimulations. Now, it is done in several ways. We are not going to talk about that now, but one of the ways is we are taking these back into the axon terminal. And we are taking these back into the axon terminal by means of endocytosis. And this axon terminal, say, we are taking them towards the cell membrane for the recycling and reprocessing of these neurotransmitters. Now, that can be done by means of this retrograde transport by the dynein proteins. Now, here is a clinical implication in this. This dynein proteins, for example, during any viral infections, for example, rabies or herpes virus, herpes, for example, zoster virus, this rabies and herpes virus actually will infect the peripheral nervous system. Okay, now peripheral nervous system, say, for example, they will infect the axon terminals, and axon terminals, say, they have to reach the cell body, because what viruses do is they actually hack the nuclear machinery of the cell. In order to hack the nuclear machinery of the cell, they have to reach the cell body. Now, what they do is they hitch a ride on these dynein proteins, and from the axon terminal, by means of these dynein proteins, they will be going from axon terminal to the cell body, where they will hack the nuclear machinery and will make more viral proteins. Because their goal is this only, they hack the nuclear machinery, make more viral proteins in order to spread that infection. Now, that viral proteins, in order to make more viral proteins, in order to hack this nuclear machinery, these viruses has to reach from axon terminal to the cell body. Now, they will be coming by means of this retrograde transport and by means of these motor protein, that is dynein. So this is another function of the axon, that is the axonal transport, and it can be anterograde transport from cell bodies to the axon terminal. It can be the neurotransmitters, the growth factors, the cell organelles, the structural proteins, and it can be the retrograde transport from the cell, this axon terminal towards the cell body by means of this dynein protein. It can be the non-functional cell organelle, it can be the neurotransmitters that are taken back to the cell body for the reprocessing and recycling, and it can be any viral infection, or it can be any virus that is infecting the peripheral nervous system, for example, the rabies and the herpes, and they will go towards the cell body by means of this retrograde transport. Now let's finish this structure and functions of the neuron with the last part, that is called as the axon terminal. Okay, here you can see this is the axon, and here is the axon terminal, making the different branches of the axon, and each branch at the end makes this bulbous structure, and that is called as the axon terminal. Okay, or the terminal button. Fine. Now, what this axon terminal is doing, it has the very important function that it is the secretory portion. Okay, this axon terminal is the secretory portion. Now, what did I tell you? That the neurotransmitters are synthesized in the cell body, and from cell body they are coming down the axon by means of the microtubules or the kinesin protein, and they reach the axon terminal, and they are stored in the axon terminal in the form of these vesicles. So, for example, here is the vesicle. Here is the vesicle, and this vesicle is containing these neurotransmitters. Okay, now they are stored in the vesicle. Now, once the action potential that was generated in the axon, that was conducted through the that was generated in the axon hillock, conducted down the axon towards the axon terminal. For example, plus 30 millivolt will reach the axon terminal. Now, plus 30 millivolt will stimulate another voltage gated channel that is present in the axon terminal, and that voltage gated channel is voltage gated calcium channel. Now, you might be understanding it now why we are taking the potential to plus 30 millivolts, because that plus 30 millivolt from axon hillock has to reach the axon terminal, and because we need that plus 30 millivolt to open another voltage gated channel, and that is voltage gated calcium channel. Now again, calcium is more outside the cell and less inside the cell. When we are opening this voltage gated calcium channel, there will be the influx of calcium ions into the cell. Okay, there will be the influx of calcium ions into the cell, and what will happen is that this calcium will trigger the process that we will talk in the next video the neuromuscular junction. Now, what this calcium will do is it will trigger the process of exocytosis by combining the snare proteins. We have the snare proteins present on this vesicle, we have the snare proteins present on the terminal membrane, and by combining these snare proteins, the calcium acts as a bridge in between. And once the calcium comes in between, these vesicles are pulled towards the terminal membrane, and then by the process of exocytosis, we are releasing these neurotransmitters into the space that is outside the axon terminal. Okay, now these neurotransmitters will make its effect on the target organ. It can be another neuron, it can be a muscle, it can be a gland. Okay, now we will talk about this in another lecture. Now here what I have to told you is that this axon terminal is very important, because it is the secretory portion. Because once the neurotransmitter is made or synthesized there in the cell body, there is no use of that until it is released out onto the target site. Okay, so that releasing of the neurotransmitter or secreting of the neurotransmitter is done at the axon terminal level. The second important thing is that it is very important for the reuptake of neurotransmitters. Okay, reuptake, because what did I tell you, that we don't want to we don't want that repeated stimulation of the target site. We want to, after it has made its action, we want to remove this neurotransmitter there. Either it is degraded there in case of acetylcholine, or it is taken by the glial cells, for example the astrocytes. Okay, it is removed there, or it is removed by the means of diffusion. Okay, one more important process that this is removed is taken back into the same neuron, and that is called as the reuptake of neurotransmitters. Now, there is a clinical implication of this. For example, there is a neurotransmitter that is called as serotonin. Okay, serotonin makes us happy. It makes our mood good. Okay, for example, the serotonin is released. Okay, it is released and it has made its action on the another neuron. They are in the central nervous system, for example. Serotonin jo hai is taken back into the neuron by means of serotonin reuptake. Okay, now here there is a protein, and that protein is called as serotonin reuptake protein. Okay, serotonin reuptake protein. Now this serotonin is taken back into the neuron. Now, in case of depressions, in case of anxiety, we don't want this serotonin to be taken back. We want this serotonin to remain there for a longer period in order to make our mood good, in order to make us happy. So what we give is we give certain drugs, and those drugs, antidepressant drugs, what they do is they inhibit this serotonin reuptake protein. We call them as selective serotonin reuptake inhibitors. Okay, we give the drugs that is called as selective serotonin reuptake inhibitors. Okay, and what they do is they inhibit this protein here, and when they inhibit this protein here, we are not taking this serotonin back into the neuron. So what does that mean? This serotonin is going to be here for a longer duration and make its effect on the postsynaptic neuron. Okay, so this axon terminal has the function of the secretion of neurotransmitters. It also has the function of the of reuptake of these neurotransmitters, and the best example is the serotonin. Okay, now that is all about the structure of the neuron, the types of the neuron, and the different functions of these parts of the neuron. I hope it made sense. If you have any query, you can ask us in the comment section. If you like this video, please hit that like button, and we will see you with another things about the neuron. With other things about the neuron, we will see you in the next video. Until then, goodbye.